I assume that's yes. All right, so good afternoon, everyone. In the next 10 minutes, I'm going to tell you a little bit about the work I'm doing in my research group to begin to address the following questions. First, we know that all living organisms can perform multiple behaviors with the same brain, but how does this multifunctionality truly work? Second, how can we design artificial systems with the ability to robustly and flexibly perform multiple tasks and change between them? Hey everyone, I'm Benson, a PhD student at Indiana University. Thank you for tuning into my talk on neural reuse and multifunctional neural networks. One of the greatest challenges in neuroscience currently is understanding how behavior arises out of the interaction between brains, bodies, and environments. This challenge becomes more prevalent when considering that living organisms have the capacity to engage in and effortlessly switch between many different behaviors. Given this dynamic nature of diverse behaviors, we are interested in investigating the ways in which neural resources are reused to perform multiple functions. Specifically, the goal of this work is to explore the extent to which neurons are functionally reused in a multifunctional neural system. All right, so within this system, there are two ends of the multifunctional spectrum. On one end, there are pools of dedicated neurons for each task. And on the other hand, there are particular neurons that overlap in task involvement. Additionally, this work is an investigation and evaluation of the tools used to analyze neural reuse in embodied systems. Also, the implications of this work extend beyond understanding living organisms to improving the designs of artificial systems to consider the robustness and flexibility of living organisms. All right, so in order to begin studying the extent of neural reuse in multifunctional systems, first, we evolve neural networks to perform multiple control tasks and then analyze their solutions. To do this, we evolve the weights and biases of a classic two hidden layer feed-forward neural network to solve three different control tasks: the inverted pendulum, the cartpole balancer, and the single-legged walker. We designed the neural network such that there was dedicated input and output for each task, but a shared hidden layer unit among all three tasks. In order to evolve neural networks to solve all three tasks, we used Harvey's microbial genetic algorithm to search for possible successful configurations. Our choice of evolutionary algorithm will demonstrate that a simple search algorithm is sufficient to evolve successful solutions. So our first step was to find a neural network architecture capable of reliably evolving to successfully solve each task individually. This step ensured both that the architecture itself could solve each task in a reasonable amount of time and that the associated task fitness functions could reliably produce well-performing artificial agents. Our second step was to evolve a single multifunctional neural network to solve all three control tasks. To do this, the fitness evaluation assessed each neural network on its performance on each of the three tasks. The resulting fitness was a product of the three individual fitnesses, confirming that solutions that performed well overall also did so on the three individual tasks. Okay, so we performed a total of 100 evolutionary runs, and we selected the top performers to form an ensemble of good solutions to analyze further. We also ensured that each of these multifunctional neural networks in the ensemble of high performers could solve each of the three tasks under a more comprehensive set of starting conditions. So here's an example of one successfully evolved multifunctional neural network under a few of these representative conditions. So first, the network is performing the inverted pendulum task, and as you can see here. So while it's performing this task, inputs are fed into the input neurons corresponding to this specific task and only taking the outputs for that task while ignoring the other outputs. Next, the network is performing the cartpole balancer task. Note that as the neural network switches from task to task, the dedicated groups of input and output neurons become active. Again, certain patterns of active interneurons emerge during the task performance. Finally, the network is performing the legged walker task. So besides noting that the singular neural network can solve all three tasks, you also may have noticed that oftentimes the same sets of neurons were active across all three of the tasks. Okay, so in order to characterize the role that each neuron plays in each of the different behaviors, we performed an information lesion for each neuron on each task. So to determine the role that a neuron plays in a task, we set the value of that neuron to be constant throughout a performance evaluation on that task, and we took note of the drop in performance in relation to when that neuron was allowed to be active. We repeated this process over a wide range of possible constant values for each hidden layer neuron, and then we kept the highest possible score. So the result is a map of the involvement of each neuron in each task. This is an example of the map of the best performing network in the ensemble. To simplify this data, we can define a threshold. So if the performance drops below 85% of its usual performance, then that neuron is involved. Otherwise, it is not. Here, each column corresponds to a different hidden layer neuron, and the 3 colors correspond to that neuron's involvement in one of the 3 tasks. We can use this simplified data set to classify neurons based on whether they are involved in one or multiple behaviors. Again, each color corresponds to that neuron's involvement in a specific task. White indicates neural involvement in all 3 tasks, and black indicates a complete lack of task involvement. For example, the mapping of neuron 2, this one right here, reveals that the specific neuron is involved in solving both the inverted pendulum and the carpal task. So what we can see here is that 7 out of 10 interneurons are reused across the 3 tasks. Finally, in order to analyze the whole ensemble of neural networks, we simplify the data even further to exclusively quantify the proportion of dedicated versus shared neural resources. So this figure is showing that all high-performing multifunctional neural networks indeed reuse most of their neural resources. Okay, so while information lesion analysis gives the ground truth results for neural involvement, this analysis is highly disruptive, experimentally challenging, and it's entirely unfeasible for all but the simplest of circuits in organisms. So a method commonly applied in the literature is the study of neural variance. In order to analyze our system using more experimentally practical methods, we evaluate the involvement of a neuron in a task using variability in neural traces. With this method, the amount of variance in the neural trace during the performance of a particular task is proportional to that neuron's involvement in the same task. When analyzing the neural variance in the same best neural network as before in the previous slide, we find that after categorizing the task involvement of each neuron, neural variance estimates correctly 22 out of the 30 possible combinations of neural task involvement when compared to the categorization of the lesion analysis. When we simplify the data further to assess the relative proportion of specialized versus reused neurons, we see that there is evidence of neural reuse among the 3 tasks, but many more neurons are exhibiting specialized task involvement as compared to the parallel analysis of the information lesions. Next, studying the variance of a neural trace is informative, but we wanted to have a more comprehensive analysis of the behavior of the neural networks to compare with the ground truth deduced from the information lesions. So our next step was to examine the usefulness of using the mutual information that each neuron has about the sensory input during a task as a method for estimating their involvement in each task. So to do this, we calculated the mutual information between each hidden layer neuron and the sensory input for all 3 tasks. We then proceeded to classify each interneuron as either contributing or not to each of the 3 tasks. And finally, we estimated the relative proportion of reused to specialized neurons as before for each neural network in the top performing ensemble. As you can see, there is a higher level of neural reuse revealed by mutual information than with neural variance. Okay. The final step of this work was to quantitatively compare the 2 methods, neural variance and mutual information, in terms of reliability at estimating task involvement. So to do this, we considered 3 different comparisons. First one over here, we measured the Euclidean distances between the normalized mutual information in each neuron for each task and the normalized variance of each neuron for each task. Here, each dot is a neural network from the top performing ensemble. So as you can see, most of the networks lie above the diagonal, denoting that the mutual information estimates of neural task involvement are closer in Euclidean distance to the information lesion scores than those of neural variability. The second plot shows the number of neural task involvement estimates that were correct for both metrics, again using the information lesion scores as the ground truth. Mutual information again reveals higher numbers of correct estimates over the ensemble of the top performing neural networks. Finally, we measure the Euclidean distance again of the ratios of specialized and reused neurons between mutual information in the lesions and between neural variants in the formation lesions. So once again, the information gathered from the mutual information is closer in Euclidean distance to the lesion scores than those of neural variants. Okay. So to summarize the contributions of this work, my collaborators and I are interested in better understanding the role of neural reuse in multifunctional neural networks. Our approach follows a long tradition in the field of artificial life of using evolutionary algorithms to search for neural networks that can perform certain tasks, and then analyzing the resulting systems using different methods to better understand how they operate. Okay, so from our work, we learned three main things. First of all, even though most of the evolutionary robotics work has stopped at one or two control tasks, there is no reason why even the most basic stochastic search algorithms shouldn't work for a larger number of tasks. Second, our main effort was not to develop a method for creating multifunctional circuits, but rather to generate some examples as a way of better understanding their neural reuse. Our analysis of the resulting solutions suggested that they did indeed exhibit high levels of neural reuse across three control tasks. So in the future, we would like to systematically examine how network topology and network size and also training regime affect these results. Finally, we learned that analyzing the amount of mutual information that each neuron has about the sensory input in each task is more informative about the role of neurons in a task than merely examining the variability in the neural traces. Also, for future work, we would like to consider more than one neuron at a time in our analysis. Ultimately, one of the directions that we would like to also incorporate is to extend the mutual information analysis to be not just about the sensory input, but also about the motor input. And that's all I have from you guys today. Thank you for listening.